Welcome to the 28th lecture of this course on environmental biotechnology, and in this lecture we will continue our discussion on the different methods used in environmental biotechnology, particularly those relevant to the analysis of microbial communities. Now, in this lecture we are going to talk mainly on the culture dependent approaches that are being used to decipher the microbial community, microbes in general which are present in any of the environmental system. And in this regard we are going to emphasize on direct versus enrichment based isolation, as we have previously learned the concept of enrichment based isolation and characterization of microorganisms from different environments. And these approaches, that is the direct isolation of the organisms as well as the enrichment based isolation of the microorganisms, will be also discussed in terms of how these organisms could be utilized to understand their environmental role, and then they can be subsequently taken for or developed for different environmental applications. We will also try to have some examples in this regard using both the direct as well as enrichment based processes. Now, as we understand that these are culture dependent approaches, so that means growth of the individual organisms present in the samples are essential and a kind of a prerequisite for this kind of approaches. So, we have different type of environmental samples. So, whatever may be our environment that on which we are trying to work in order to understand the microbial communities, and we are trying to answer the 4 questions that are actually the integral part of the microbial communities with respect to environmental biotechnology. Now, from any environmental sample we can go for a direct isolation based approach, and we can also have alternatively the enrichment based isolation approach. In both these approaches, the essential outcome would be the bacterial strains as they are pure culture. So, as I have already discussed, direct isolation basically refers to any kind of procedure where the environmental samples, whether it is a solid, semi-solid or liquid sample, it is diluted in appropriate medium and then added on different microbiological media, solid media in particular, so that the individual colonies are obtained directly. So, the samples are diluted and added directly onto the top of the agar or within the agar in case of the anaerobic organisms, and it is expected that the microbial load or the concentration of the microbial cells in different environmental samples like the wastewater treatment sample, like the activated sludge, like any other environmental samples, will be too high in the order of 10 to the power 6 or 10 to the power 8 cells per gram or per milliliter of the samples. So, we need to have actually a dilution, and those dilutions are made using a sterile medium or is kind of a suitable buffer, or sometimes the sterile saline, so that the osmolarity of the organisms or osmolarity of the environment is maintained, and we are able to have individual colonies appearing on the plates or appearing on the anaerobic agar, and we isolate those individual organisms and then we characterize them. In case of enrichment based isolation, as we already discussed, certain enrichment conditions are created based on the substrates, the conditions, those are relevant to for a particular sample. I will try to explain the enrichment based procedure in little more detail after some time. So, basically whether we are using the direct isolation technique or enrichment based isolation technique, we will be having some pure culture bacteria eventually with us. Now, this culture purity is obtained through repeated subculturing. So, these are standard microbiological procedure and we are not going to discuss much on that. So, eventually the outcome is the isolation of bacterial strains as pure culture. So, they are pure. So, they are not mixed with any other organisms. Now, as soon as we have those pure culture or pure organisms, maybe 10 in number, maybe 20 in number, maybe hundreds in numbers. So, depending upon the sample and depending upon our research objectives, we can try to isolate as many number of strains as possible. And eventually those organisms, those individual colonies, those individual strains are subjected to characterization, and this characterization basically includes the metabolic and ecophysiological characterization. Now, we have, for example, if we have 20 bacterial strains isolated as pure culture from a given sludge or given contaminated environment or any kind of other environment where we are trying to understand the microbes play in terms of their role in environmental process development. This metabolic and ecophysiological characterization is a kind of a very important and critical term or critical phenomenon, because what are the metabolic characteristics or ecophysiological characteristics those we are going to select is actually defined by what is the environment, and within that environment what are we trying to do. For example, if we have a petroleum refinery waste, for example, or a petroleum oil contaminated waste, what is our research objective? What is our objective in terms of environmental biotechnology? Are we looking at the remediation of the waste, petroleum waste, or are we looking at the production of some valuable chemicals or production of certain gases like methane from this waste material, or we are looking at a combination of all these? So, if we are looking at, for example, the remediation part of the waste, then the kind of metabolic and ecophysiological characteristics which will be required to be assessed with the isolates will be somewhat very precise, very defined with respect to their degradation abilities, degradation of the different aliphatic and aromatic compounds, their ability to withstand different heavy metals, etc. But if we are targeting converting this petroleum waste into methane, for example, then perhaps we have to see that how specifically the organisms which are responsible for, or the processes which are responsible for methane generation from the hydrocarbons which are present there need to be assessed. So, these are highly again process centric. So, what exactly we are trying to do, what is our objectives, what is the goal of with this particular environmental sample? Based on that, the metabolic and ecophysiological characteristics are often selected. Although there are certain kind of norms or dogma that we generally try to follow with respect to certain cases, that if we want to or if we intend to describe some of the isolated bacterial strains in terms of their novelty, that how a particular strain is novel or its detailed taxonomic description, then perhaps we have certain set of techniques that I will give one example to understand that. Otherwise, the selection of these metabolic and ecophysiological characteristics are highly the process driven. What is the process, what is the research objective, what is the technology that we are trying to develop, based on that these characteristic properties are selected. The next one would be with respect to these isolates those are obtained is the identification of these strains. Suppose we have 10 or 20 bacterial strains, so we need to actually identify them, and in today's point of time the molecular identifications are generally practiced because that is more sensitive and that is going to give us a substantial information which will be useful in many sense. So, the molecular identification is basically done using a particular gene which is called the 16S ribosomal RNA gene, which encodes basically the ribosomal RNA. Ribosomal RNA is a part of a ribosome. So, essentially this is a very well known molecular marker we called for delineation of the taxonomy of the prokaryotes like bacteria and archaea. In case of the fungus and other eukaryotic organisms like microalgae etc., we may need to change this gene to 18S ribosomal RNA gene, because 16S ribosomal RNA gene is more specific to prokaryotes, whereas 18S ribosomal RNA gene would be suitable for the eukaryotic organism. So, the identification of these organisms can be done. So, whatever isolates are obtained, the isolates will be characterized and isolates will be identified. Now, this information would possibly help us to answer the first two questions that we originally proposed, that what are the organisms present there and what are their capacity or what are their potential in terms of the metabolism or metabolic functions. Now, I will elaborate upon that how do we actually go with respect to the direct versus enrichment based isolation of microorganism, particularly when we try to decipher their environmental role and move forward with respect to their applications. So, for example, if we look at a sediment which is collected from a kind of a natural habitat, and here you can see that the sediment sample is done by in a group of scientists who are working on a lake sediment possibly, and here is the very standard sediment collection procedures are followed. Now, once we have the sediments are subjected to some kind of studies, and one of the, in this case, one of the goal of the study is to decipher the microbial communities, and then from that the environmental biotechnology relevant questions are to be answered. So, as I mentioned earlier, that one straightforward approach would be that the culturing the pure culture bacteria. Now, we know already that only maybe 1% or less than that of the total microbial populations residing in any environment are cultivable. Nevertheless, since there are large number of microbial species present, like I said, around 10 to the power 4 to 10 to the power 5 species, different species are there, and total number could be up to 10 to the power 8 cells or 10 to the power 9 cells per milliliter or per gram of soil. So, it is a huge number of cells and a considerably high number of species are there. So, even if 1% is cultivable, it means a substantial number of cells or strains are still cultivable. Although, with respect to the entire population or entire community, that may not be very significant, but still it is some number, some cells. So, these strains might be providing us some important information, might be useful, and always culture based techniques are very well developed for a general purpose. So, we can go for a culturing of these organisms, keeping in mind that they are only going to give us some information about the environment or environmental process. It is not the total picture, that has to be in our mind. Alternatively, we can have the enrichment based methods, and these enrichment based methods are controlled by different substrates, different conditions, as dictated by the conditions or as driven by the research question that we are trying to answer. So, for example, as I mentioned, for hydrocarbon degradation, enrichment setup will be specific, whereas for heavy metal removal, the enrichment will be different. For methane production or other gas production or other metabolite production which are biotechnologically relevant from a waste, the enrichment could be different. So, the enrichments are very much purpose driven, that what we are trying to look at a particular within a particular sample. So, anyway, so if we have the enrichments, then we have the enrichment cultures. These enrichment cultures are necessarily or mostly found to be a multi-species assemblage, because out of all the organisms present there, possibly some of them, obviously the sum is more than what we can get out of this culturable pure culture bacteria, would be more during enrichment procedure. It has been found that most of the cases, the enrichment based methods are, if the enrichment is set up, enrichment is done with the proper consideration of the facts that actually refers to the real environment and the environmental processes which are going on in a particular setting. So, the enrichment of multiple organisms would be possible. Now, here these organisms are not individual here. They are individual, but they are growing together within the enrichment container or reactor or enrichment flask or a tube, wherever we have set up the enrichment. These color balls represent actually a different species, and these different species could grow together. Now, there would be a very interesting point. and that compared to the total community, if we just imagine with this cartoon that the total community was having many more species, only a very few could be grown on a pure culture, but some more numbers could be grown as mixed multiple species growing together within the enrichment. Now, now if we take this enrichment culture, which is a broth growing in a test tube or a conical flask or a reactor, whatever it is, we can actually try to isolate some of these organisms as pure culture. So, what we will do, we will take some of it and we will add on or the spread onto the solid agar medium, as we have possibly done for the pure culture isolation procedure using either aerobic or anaerobic condition, and eventually some of these enriched organisms might grow as pure culture. So, may not be all. Why not all? Because some of them might be requiring strictly the interaction between the species themselves, and if we try to segregate as to obtain the pure culture, requirements are not satisfied, because the mutual interaction between two organisms are missed when we try to segregate them physically during the spreading or high dilution. So, eventually we may lose them. So, some cells might grow depending upon what type of system. So, there are examples where we are unable to actually grow any organisms as pure culture from the enrichment. On the other side, there are examples where a number of organisms out of the enrichment culture were successfully grown as pure culture as well. So, it depends upon what type of environment and what are the enrichment conditions that we are trying to deal with. Now, these organisms which we could grow as pure culture from the enrichment based technique could be segregated, could be picked up, subcultured and then purified, and then we try to characterize them, and this is called we get them and characterize them, and that is actually a question in terms of how do we characterize them. So, I was referring to these characteristics, determination of these characteristics, that what are the parameters that we are supposed to select. So, these are again connected to what is the research question that we are trying to answer. So, depending on that, based on the research question or the process that we are trying to decipher, these characteristic parameters will be defined. Now, it is also obvious that some organisms out of this enrichment culture will remain uncultivable. They will not respond, as I was mentioning that none of them could be, or some of them could be grown in pure culture, but others may not grow as pure culture. So, they remain uncultivable. So, we do not know about this, because as we try to plate on agar medium this as inoculum, we will be getting most of the cases a few colonies, but we do not know that whether others are there. So, we have to take it like a fact that there could be some bacteria surely or archaea who are not growing in the medium that we are using or the condition that we are using. So, if we believe that in most cases it is found to be true that within an enrichment culture there are again more than 80% or 90% organisms are there who are not cultivable. So, we can actually decipher and characterize them as well, and we need to. So, in this case, what are you going to do if we have some kind of issues like that we have enrichment culture and we want to know what are the organisms those are enriched, what are the functions they are carrying out, and how they are going to be helpful for any environmental biotechnology process. So, one idea could be using the improved cultivation strategies. That means, by looking into the literature, by looking into the process, looking into the conditions, we need to think that how we can help the microbes to grow better. What are the substrates, what are the conditions that we should provide so that more number of organisms can be grown. And another more straightforward approach would be cultivation independent approach, where we have to rely on certain other methods that maybe I will be discussing in our some next lecture. They are the cultivation independent. So, no growth based strategy or agar medium based strategy will be required. We can take the enrichment culture and straight away try to characterize these organisms following different strategies. Now, the setting up of this enrichment, that is a very interesting part, because how do we create the condition, how do we set up the real environment where the specific group of organisms will be enriched. So, ideally it is considered to be a process centric. What is the process that we are trying to investigate, try to study, try to exploit possibly for the environmental benefit. So, one way of looking at it is the resources that we can provide, like the carbon, nitrogen, phosphorus, other nutrients individually or in a mixture, different electron donors, different electron acceptors, or any other specific substrates which are relevant for a particular environment. For example, pesticides, herbicides, different type of heavy metals, they can be often used to test that or allow the enrichment of organisms which can be which are able to actually sequester those heavy metals or degrade those pesticides or herbicides or something like that. And that is also true possibly for the plastic or microplastic degradation or remediation process related enrichments. The second is the conditions. So, resource is not enough, the condition is also very important. So, the conditions could be also varied for different enrichments, like the redox conditions can be varied, including the oxidizing conditions to the moderately reducing conditions to strict reducing conditions. The pH can be varied, the salinity can be varied, pressure can be varied, temperature can be varied. So, there can be different type of variations, different type of systems that we can prepare, and we can actually incubate our sample under that condition in order to enrich a specific type. Like if I want to isolate some extremely halophilic bacteria or archaea, or if I want to isolate or see with respect to high temperature, what are the organisms which are able to withstand high temperature. So, it is the best approach would be that set up an enrichment culture at high temperature and use the carbon and nitrogen sources and other electron donors and electron acceptors which are relevant to the real environment. So, whatever is available in the real environment, try to keep them, try to put them. So, how do we put them, or how do we prepare our medium for this case? So, for that we need a detailed chemical analysis of the waste material, and then based on that detailed characteristics of the waste material, we have to prepare our medium, and that medium has to be incubated at temperature, higher temperature. Suppose I want to see if there are any organisms capable of growth at 50 degrees centigrade while utilizing the organic matter and the nitrogen, nitrate or ammonia present in my wastewater. So, I need to then incubate my sample at 50 degrees centigrade and with occasional subculturing. So, if we maintain that temperature, so with frequent subculturing, we will get some of the organisms enriched. There could be possibility that none of the organisms are enriched, but there is high possibility that many organisms are enriched. And overall, this enrichment is again a cultivation based method, because you are allowing the organisms to grow within your laboratory setup. So, in a nutshell, this resource based enrichment setup, condition based enrichment setup, and a combination of multiple factors. Combination of multiple factors meaning that we take into account both the resources as well as the conditions. Then we set up the enrichment, so that we are close to the real environment. Only thing is that instead of a real environment, we have taken the sample and we are trying to grow the organisms in the laboratory based on our preferred substrate. The substrate that we intend to see that whether these substrates are, these conditions are okay with these organisms, or organisms are rather okay with this. Now, we will discuss a bit of this analysis of the enriched samples. That once we have this enrichment done, maybe as I said that we have this our enrichment, we call enrichment culture. So, this enrichment culture is done. So, this may be based on different substrate or different condition or both. Now, once we have this enrichment culture, so this culture could be in a tube or in a flask where we have this so this is the culture where my organisms are surviving, but I do not know about them. I want to know. So, what do I do to understand that what is going on in this culture? So, what I am supposed to do? I have to analyze two things. One is the medium. The medium has to be analyzed, and the second is the cells need to be analyzed. Why do I need to analyze my medium? Because I have my substrate there. Suppose I have added the crude oil in a petroleum waste to see that whether I can actually enrich some crude oil degrading bacteria, or if I want to see whether I have some methane producing bacteria. So, in that case possibly even we have to take the gas sample as well. So, this is called the headspace that we need to actually use. So, we have to sample the headspace using a headspace sampler, and obviously for that we need to have a kind of a airtight incubation if we are planning to have a kind of gas production in that. So, the medium, the headspace, all, so there could be the products. Products means the gases. Now, the medium is analyzed not only for the substrate, but also for the products. Products in the sense the products which are produced out of the substrate. So, we have added for example the hydrocarbons. So, we have added suppose we have set up an enrichment for the hydrocarbon metabolizing organisms. So, we have the hydrocarbons added into this culture. So, after maybe 20 days or 21 days or 1 month, the hydrocarbons are supposed to be converted into something. So, what they have prepared? So, they must be formed some metabolites. So, those metabolites are to be checked that whether those metabolites are there, what are the metabolites present there. So, by knowing that what are the metabolites present there, we will be able to assess that how these organisms are actually utilizing the substrates. And also if we analyze the headspace gas, then we will be able to find out whether hydrogen is produced, carbon dioxide is produced, or the methane is produced in that, and we will have some idea about what are the possible metabolic processes going on there. And now eventually the cells. Now again for the cells we have the very streamlined procedure that to characterize them. Now characterize them in terms of their metabolic properties, characterize them in terms of the genes they have or the enzymes they are producing, or delineate their taxonomy. So, when we characterize the cells, so we may not be able to isolate individual cells. So, it may be sometimes it is the pure culture that we get, sometimes it is the mixed culture, but sometimes it is the total population which is enriched. So, sometimes it is the pure culture from this. So, we are able to isolate some pure culture, or we have enriched a mixed culture, or what I said earlier that some organisms or many organisms will be not cultivable even from the enrichment culture. So, the total populations of the enrichment culture can be assessed by some method, and we will discuss about this method. This will be done by the cultivation independent method. That we will discuss later. So, once we have the enrichment, do not think that only the organisms will be isolated and characterized in order to know the process they are in that they are involved. We need to see the medium, we need to see the headspace in order to understand what are the things going on actually. So, some enrichment methods we are going to have some description about some standard procedures that how we generally go with this enrichment system. So, these are some of the examples which are very well cited in the literature. For example, for the phototropic and chemolithotrophic bacteria. If we want to suppose enrich phototroph and chemolithotrophic bacteria, for phototrophic bacteria which requires basically the light and the main carbon source is carbon dioxide. So, we can have oxic incubation as well as anoxic incubation, because there are many bacteria who actually do not require or do not like oxygen. So, they are anaerobic, while some organisms are aerobic. So, we can have aerobic incubations, we can have anaerobic incubations, and we can have nitrogen or nitrate as the nitrogen source. We can have normal room temperature or elevated temperature in order to look at the or enrich the thermophilic organisms, and there are certain literature available which suggests that under these conditions what are the organisms which are expected, because with a light based phototrophic processes, the organisms which can be isolated or enriched are mostly the cyanobacteria or the thermophilic cyanobacteria, and a number of environmental samples and environmental other things are being subjected for this kind of enrichment. Similarly, for anoxic incubation, we can have, since it is anoxic, we must provide the electron acceptor. So, electron acceptor could be iron, could be nitrate, could be sulphate, and as electron donor we often provide hydrogen, or sometimes the organic materials, organic acids are provided, sometimes reduced sulfides are also provided, and these are the type of organisms like purple non-sulfur bacteria or green sulfur bacteria, these are generally enriched. Now, for the dark chemolithotrophic bacteria which actually do not require any kind of light sources, the electron donors are again for aerobic and ammonia to nitrite to hydrogen, etc. And for the anoxic incubation we can have the sulfur or other sulfur compounds, hydrogen, Fe2 plus, etc. And for anaerobe we can have the electron acceptors which are mostly found to be nitrate, and preferred is the nitrate, and for aerobe it is the oxygen. And we can expect that the organisms which are enriched are different type of ammonia oxidizing bacteria, nitrite oxidizing, hydrogen utilizing bacteria, as well as some of the organisms which are found to be thermophilic, because if we set up the enrichment at high temperature, but they are all capable of doing chemolithotrophy. That means they are utilizing these inorganic resources as electron donor and inorganic material as electron acceptor, and they are capable of functioning. So, in any environment if we want to set up or want to know who are the chemolithotroph, anaerobic chemolithotroph, then perhaps an anaerobic incubation with this kind of substrates would be beneficial. Similarly, the chemo-organotrophic bacteria, for which again the aerobic with a number of substrates which are reduced organic carbon, and if it is aerobe then the oxygen as electron acceptor, and the nature of organisms that can be isolated are very broad, a large number of taxa can be isolated. And similarly, if we have an anoxic incubation for this heterotroph, we will have the organic substances, but oxygen will not be there, but instead of oxygen we will have specific electron acceptors which will substitute the oxygen, and based on the electron acceptor, as we already studied, that different type of organisms again will be enriched. And then similarly, the fermentative organisms can be also enriched. In case of fermentative process, we won't add any electron acceptor, and only the electron donors are used, which are the organic compounds, and we know that by definition of fermentation is this the endogenous electron donor, electron acceptor, I am sorry. So whatever electron acceptors will be generated during the glycolysis process, like the pyruvic acid for example, that will be utilized and the organisms will be enriched. So again, a large number of organisms belonging to the taxonomically very well defined Clostridium and other organisms will be enriched. So now I will quickly give you some examples of these enrichment based strategies based on our own research work. So in one of the studies that we tried to actually enrich and characterize the hydrocarbon degrading bacteria from petroleum refinery waste, and our research goal was to use these bacterial strains, the pure culture bacterial strains that we aim to obtain as bioaugmentation agent in the in situ bioremediation process. And what we observed that when we adopt this enrichment based process, a number of different organisms, these are the name of the taxa or the genus. So as you can see that a number of species member belonging to different genera are opted. Now, here is a very interesting fact, that when you go for the direct isolation, like as I mentioned, the sample was taken and diluted and spreaded onto the agar medium, we found that it is basically Pseudomonas and the Bacillus who could grow as individual colony, and we are able to isolate them. So, it is basically the only two genera, Pseudomonas and Bacillus, were there as cultivable member when we perform the direct isolation. But when we enriched the same sample with crude oil for 1 month, and then we isolated the bacterial strains as pure culture, you can see there are a large number of different species like Granella, Enterobacter, different members of Burkholderia, Pandore, Microviguli, Brevundimonas and Cocuria type of bacteria. And it is interesting that the members of Pseudomonas and Bacillus species were not so well represented when we have this enrichment based procedure. So, that clearly actually defined that how direct isolation based procedure and enrichment based isolation procedure could lead to discovery of or the isolation of organisms which are entirely different and which could be more useful to understand the environment relevant process. As you can see in this next slide, that if we now look at the ecophysiological studies that we referred as earlier, that once the isolates are obtained, we need to do their metabolic profiling and ecophysiological studies. So, here I am showing you that data, that how all these individual bacterial strains which are pure culture are subjected to different pH, different temperature tolerance, different sodium chloride concentration tolerance, different heavy metal tolerance, as well as utilization of different hydrocarbons as an individual substrate. Like the first one is the BTX, benzene, toluene, ethyl benzene and xylene, which is a very common